So we're going to go over some notes on Chapter 6. Chapter 6 has to do with metabolism and enzymes. Alcohol dehydrogenase occurs in the liver and it breaks down toxic alcohols. So when you drink alcohol, it breaks down these toxic alcohols, your liver does, but it does it at the expense of the liver function and energy metabolism. And when you have cirrhosis of the liver, when people have problems with that, it doesn't necessarily mean that they ever drank alcohol because cirrhosis of the liver can also be caused by other factors such as poor nutrition. So the first law of thermodynamics is that energy is neither created nor destroyed, and so energy is transferred instead from one point to another, but not created or lost. So in the second law of thermodynamics, the amount of energy available to do work is going to vary. So you have a term called entropy, which is the measure of dispersal of energy, and this can happen with two atoms. And the entropy of two atoms decreases when a bond forms between them, and that would be considered an inorganic reaction. And in an inorganic reaction, you're going to put a net amount of energy into the reaction. And of course, you should know that energy is always escaping as heat, and energy flows on the earth from the sun to producers. So the sun gives energy to the plants, and then the plants we consume as consumers. So that flow of energy flows through the entire food chain. And then, of course, living things need a constant energy input in order for you to be able to sustain life and go to school and play basketball and play sports and football and golf and all of these things. You need to be able to have energy inputs and outputs. When you have energy conversion, only about 10% of that energy that you eat from your food goes toward building body mass because most is lost due to energy conversions. So it's not 100% energy efficient at all. You're constantly taking in energy and releasing energy. So energy in and out all the time. You lose and gain energy all the time. So in order to maintain energy, you have to eat food and have your blood glucose level to a certain level, and then you're going to use that energy to do your daily activities like just get up in the morning and get ready for work like this lady in this picture, or to play basketball or sports or whatever it is you need to do. So energy is constantly being replenished and you're constantly using energy. Even when you sleep, you're going to burn some calories and use some energy. There's something very important in your body called activation energy that occurs. So let's talk a little bit about activation energy. Activation energy is going to lower the amount of energy it takes to do or have reactions rather. So let's discuss some terms. ATP, of course, is adenosine triphosphate. You've already learned that in class. And you have ADP, which is adenosine diphosphate. And if you add another phosphorus, that will turn into ATP. And if you take a phosphorus away, it'll go back to ADP. And that occurs in your body all the time. So ATP is a nucleotide with 3 phosphate groups because it's the tri part, and ADP would have 2 phosphate groups because it's di. And so there's transfers. Basically, what happens is when you transfer those phosphates, then you get a difference between either ADP or ATP. So when that process happens, when you're adding a P or you're taking away a P, it's called phosphorylation. So phosphorylation, if you look at the word, sounds like phosphorus. So you have phosphate group transfers all the time. ADP will bind an extra phosphate or release one. And when it binds it, it's called an inorganic reaction. And that replenishes that extra ATP to make that from ADP to ATP. You're gonna bond an extra phosphate so that you can change back and forth. So when phosphorus is added or taken away to convert ADP to ATP or vice versa, you have phosphorylation. And sometimes people call that the ATP ADP cycle. And then over on the right, you're just going to see a little diagram that shows you what has to happen in order for cellular respiration to take place, which occurs in your mitochondria in your body, which is not like just breathing in and out. It's not that type of respiration. It's respiration inside your mitochondria that take energy from the food you eat and convert it to that ATP to allow energy from your cells to occur. That's the simplified version of it. So we've discussed enzyme substrate complex in class, and that also has the terminology or the term lock and key mechanism and induced fit model. And it's called enzyme substrate complex because the enzyme and the substrate fit together in the active site. So we've talked about the enzyme looking like Pac-Man and the substrate looking like a piece of pizza that fits into Pac-Man's mouth. And then the active site would be Pac-Man's mouth specifically. So we discussed that in class, and you should have a drawing in your notebook. So enzymes we've also discussed in class are catalysts, which we know will cause a reaction to occur faster. And most enzymes are proteins, and some of them are RNA. And enzymes are not changed during the reaction. They just fit together differently to cause that reaction to occur or cause that reaction to stop occurring. So whenever you have enzymes in your body, they're necessary because otherwise you would need a huge amount of energy to cause reactions to happen, or either they would just happen really slowly. So they lower what's called activation energy to allow that reaction to occur. So enzyme-mediated reactions, you have the substrate, which is the reactant added upon by the enzyme. That's the official definition, and that's the piece of pizza. Then you have the active site, which is the location where the substrate's going to bind and reactions happen, which is Pac-Man's mouth. And then you have the fact that the active site is going to be a certain shape and size and polarity and charge in order for that active site to recognize and accept that substrate. So that's important in enzymatic reactions inside your body. And then the concentrations of enzymes and substrates in your body can cause or stop reactions to occur. And of course, you have feedback mechanisms which can adjust enzyme production or activate or inhibit the enzyme, which basically means feedback mechanisms are when the easiest explanation is when something goes up in your body, some type of substance, and that causes something else to go up inside your body. So the two increases, one increase causes a subsequent increase. That's a positive feedback mechanism. And when you have something that increases in your body, which causes a decrease in your body, and that's a negative feedback mechanism or loop. We don't really go into that a lot right now, but we do whenever we get into the human body, the second part of the class. So sometimes you're going to hear the terminology allosteric site. So molecules combine to allosteric sites to activate because sometimes you need those enzymes to be activated and sometimes you need enzymatic reactions to be inhibited. So allosteric sites can help for both of those. So if you look at this picture, you'll have at the bottom and the top, you'll have arrows that go toward the right. And that just shows you a little pathway of allosteric sites and shows you that there's more than one location where molecules combined. And sometimes the molecule or the substrate combined to the enzyme, and sometimes it can't. So in enzyme binding and active sites. If you look at this picture, this is just once again an example of an enzyme substrate complex and an active site that it shows you. So enzyme binding and active sites brings on something called the transition state, and this is what happens. You have this transition state that's going to help your substrate to bind, and then it's going to orient the substrate to fit into the active site because as you look at this, it's just like a puzzle piece. If you flip the puzzle piece the wrong way, it's not going to fit into where it's supposed to go. So you have to flip it and orient it correctly. And whenever you do attach the puzzle piece, sometimes the puzzle piece can be barely attached and not stay there very well, and sometimes it can be very firmly attached. And so the transition state helps to bind the substrate to the enzyme and also helps to get in the right position for it to bind. And of course, when this binding occurs, there's all kinds of things we've talked about in class that can influence this. We talked about temperature already, and we talked about pH, but we didn't mention salt yet. But salt concentration, sodium chloride, can affect that binding as well of the enzyme, or the substrate to the enzyme. When you look at endergonic versus exergonic reactions, endergonic is going to be energy in, and you're going to have a net input of energy, and exergonic is going to be energy out, net release of energy. So if you look at ender, that sounds like energy in, ender in, and exer is going to be like exit or release or go out. Metabolism is any chemical reactions inside your body. So metabolic pathways are any of these energy-mediated reactions that we've talked about already. So in order for you to build new cells and have repair and have things work in your body correctly, you're going to have metabolism occur and metabolic pathways. So to build cells, rearrange cells, and to take the food that you eat and build it up for energy, then you have to have anabolism or anabolic metabolism, which is when you build molecules. So you take the food you eat, put all that together, and you're going to build cell growth, you're going to build cells, you're going to build anything that you need in your body. And then catabolic or catabolism is when you break molecules. So for anabolic, think anabolic steroids are what people take to build muscle, not break down muscle. And so these cyclic pathways help to have all this energy conversion and have your metabolism work properly. So oxidation reduction reactions can occur, and so you have molecules that give up electrons and are oxidized, or molecules that were reduced by accepting electrons. And of course you have coenzymes that can accept in redox reactions, also called electron transfers. So these coenzymes that help oxidation and reduction occur. And it's important, all these are important in electron transfer phosphorylation, which we'll talk about later on. If you look at the bottom, there's a picture of a substrate and an active site, and then you're going to have a coenzyme there that's also going to help that substrate to bind to the active site. So it's not just an active site that you have all these other additional types of little helpers is what I'll call them. So when you have electron transfer chains, I want you to think of it as like a stair step. So you have to have a series of these redox reactions that occur in order to bring on change or to increase the energy level from a lower level to a higher level. So this is what occurs for and molecules then can release energy. So it occurs in small little steps that equal bigger changes over time, but it happens in small little increments. Just like when you start up and you get up out of the bed in the morning. You don't just get up and probably run laps around the house. You get up and you just move around and mull around really slowly, and then you have more energy. So that happens over an extended period of time slowly. In an electron transfer chain, you have all these little bitty steps that equal to a bigger change. And you're going to see a video link on this page, so you need to watch the video about electron transfer chain. So temperature, pH, and salinity can affect the changes that occur in enzymes. So the higher the temperature, the greater reaction. But at too high temperatures, too many extremes can cause the enzymes to denature. And we've talked about that in class, and we've talked about how that active site can change shape if it gets into too extreme of an environment. And so lower temperature, that's going to have less reaction. And of course, the denaturing can occur if you get way too low temperature. And then each enzyme is going to have an optimum pH that it works at as well. And so your body's going to have to stay at that pH for that enzyme particularly to work. And most enzymes like it between about 6 and 8 pH. And the other thing that's going to affect enzymatic reactions and functioning is going to be salt levels, which affect those H bonds or hydrogen bonds that hold the enzymes in that three-dimensional shape that they have. So temperature, pH, salinity, salt, all of those things can affect enzymatic reactions. So we discussed earlier that there's more than just the active site that aids in the substrate binding to the active site. You have something called cofactors and coenzymes. So cofactors are atoms or molecules that are other than proteins that allow for enzyme function. And an example is iron atoms in catalase. You also have coenzymes, which are organic. An example is like vitamins, and these can be modified during reactions. And those deliver electrons to electron transfer chains for ATP synthesis. So it delivers electrons to that stair step of electrons to allow small amounts of energy change to occur to equal to a bigger change like I just discussed in the last slide. So you're going to hear a lot of discussion about antioxidants, but this slide is labeled catalase. So let's talk about what we know about catalase already from your class. So you used potato catalase, which is present in potatoes, and you smashed up the potatoes and you added peroxide. And so in the presence of peroxide, catalase bubbled up. Some of you had a small reaction and some of you had a very pronounced reaction. So catalase is certain types of catalases are antioxidants and they neutralize free radicals. And you've probably heard a lot about antioxidants and food and marketing for food having antioxidants in it. And the reason they talk about antioxidants is they're supposed to help you rid your body of these free radicals, or at least neutralize them. So free radicals are these atoms or molecules with unpaired electrons that can actually attach and cause biological molecules to be harmed. And so catalase aids in transition states or enzyme function. So when you have catalase, then it can actually aid in this free radical issue that you may have. How do enzymes work? You know that enzymes speed up reactions and they fit so the substrate fits into the active site. But you also know that reactions in the body could be too slow without the help of these enzymes. If you look over on the right, we discussed, the reason why I use this picture is we discussed salivary amylase and how as soon as you put food in your mouth, the salivary amylase is an enzyme that starts the digestion process as soon as the food enters your mouth. On the right, you have this dog, and this is a picture that I found on the internet, and I'm assuming it's talking about Pavlov's dog, because Pavlov trained his dog, and every time he'd ring a bell, he would present food, and then eventually he could just ring the bell and not present food, and the dog would start, his mouth would start salivating, and he would be thinking he was going to get food. So the reason I have this picture here is because salivary amylase is a type of enzyme, and there'll be a video for you to watch here. So metabolism and enzymes, they're going to work with constant reactions. So basically, you're always having enzymatic reactions and metabolism within your body. Of course, there has to be energy, and it has to go in a certain sequence and fashion. It just can't be sporadic, otherwise your body wouldn't be efficient at all. And there are enzyme-mediated reactions that occur in your body, so you have to have those enzymes. The concentration of materials matter, so you have to have enough. Conversions are always occurring. Your body is going to have to dispose of materials not needed inside the cells, so it's going to have intergonic and exergonic reactions. It's going to have energy in, it's going to have energy out, but it's also going to have to get energy in, and it's going to have to get waste out. So it's just like you eating food. You eat food, and you also have some waste products that come out. And so you have to have inside every single cell in your body, every cell in your body has to have that ability to release that waste as well. Otherwise, you would have waste products built up in the cells in your body, which wouldn't be good. Enzymes speed up reactions, so they're catalysts. And you can shift cell activities fast from one function to another if you have enzymatic reactions occurring inside your body. Bioluminescence, one of the most interesting slides in this entire PowerPoint, in my opinion. So organisms can make their own light. Lightning bugs make their own light. All kinds of ocean critters make their own light. And I'm going to post a video for you to watch, and you can go out on the internet and look at all kinds of bioluminescent videos. So these organisms make their own light, and they do it with enzymes. And what they do is they convert chemical energy in luciferins, which is a type of enzyme, to light energy. And so you have luciferin plus ATP,
yields luciferin plus ADP plus P. So they're going to do or undergo that chemical reaction, and that's how they're going to make their own light, which is amazing. They use some of these enzymes that they extract from ocean critters in the medical field. When they run dye through you, they will collect dye, or have in the past, this dye or this light. They also do research on lightning bugs. Around Wythe County, they've actually had lightning bug collections where they will collect up lightning. So light versus dark reactions in plants. You have a packet in your packet of notes that are printed and also in the PDF on the website. You have a packet that says light versus dark reactions, and I'm going to post a video, but you need to complete the worksheet. So you look at the first page and you read it, and then use that page to complete the next page that has a bunch of letters on it that leaves blanks out. So if you'll complete that, that'll help you with light versus dark, and we'll have some videos and some more explanation for that on another PowerPoint.